So welcome to required practical number 6. We're going to investigate the effect of light intensity on the rate of photosynthesis using an aquatic organism. So we're going to use some pondweed today, which we have in our tank. Also, you will need a meter ruler, a lamp, a stop clock, some scissors and a paper clip, a beaker with water in, a boiling tube with water in, and a thermometer too. So for stage 1, we're going to start off by collecting our sample of pondweed. So you'll need your scissors and paper clip. I'd like you to go into your tank. This is what pondweed comes like from the fish tank shop. I'd like to collect one strand about the length of a boiling tube and cut it at a 90-degree angle, like so. Now you can do that underwater. I'd like to take your paper clip and weight down the other end of your pondweed, so the cut end here, and then place your paper clip onto the pondweed there. Okay. Now I want you to take that and I want you to place it inside the water, inside your boiling tube. So I have one of those done earlier. Now you'll see there's my cut end, and you'll see my paper clip at the bottom. I want you to place that into your beaker of water. Now the reason why we do this is because the lamp will give off some heat energy, and we don't want the heat energy to affect the rate of photosynthesis. We just want the light to be affecting the rate. Okay, so we control that using our water bath. Now what I want you to do is take your meter ruler at 0 centimetres in line with the lamp, and I want you to place your pondweed starting at 50 centimetres, so the edge of your beaker on 50 centimetres away from the lamp, and switch your lamp on, and I'd like you to leave your pondweed where it is for 2 minutes and do nothing. And I've got one of those set up. So this has been at 50 centimetres, bubbling away for 2 minutes. Now I want you to pop in your thermometer just to be checking that that temperature is monitored. Okay? So I want you to focus in on the cut end of the pondweed. So we're looking, oh, there was one. So I want you to look for bubbles coming out of the cut end, and they will be oxygen bubbles, proving our pondweed is photosynthesizing. Now we're going to be counting, there you go, counting the bubbles, how many are produced in 1 minute at this distance. So we'll do that now. You can see it's going quite slowly, but they are coming out there, the oxygen bubbles. So let's imagine we've done that for 1 minute. So we have some results here. At 50 centimetres, our first trial, we got 17 bubbles in 1 minute. Now I'd like you to repeat that 2 more times. And I'd like you to calculate a mean. So remember how we do that, guys. We add up all our results, so it'd be 17 plus 19 plus 22, give us our total. And I'd like to divide that by 3, because we did it 3 times, which will give us 19. So here's some results I've collected earlier, and you'll see I've got all of my means calculated from 10 centimetres all the way through to 50 centimetres. Okay? Now we're also going to have to do light intensity at each distance, because that's what we're investigating. Now there's a calculation we need to do for this. So we're going to use the inverse square law to calculate light intensity. So let's imagine we're doing for the lamp 10 centimetres away. We're going to do 1 divided by 10 squared, because it's 1 divided by distance squared. So you'll see actually we can do 1 over 100, gives us a light intensity of 0.01 arbitrary units. One more example. There you go. For 20 centimetre distance, we have a light intensity of 1 divided by our distance squared, or 1 divided by 400 gives us our arbitrary units there. Now you'll see here, I've popped down all my calculations for each distance, and then we're going to use these 2 columns and plot this on our graph. So we'll go over to our graph. Okay. And you'll notice I've got light intensity along the bottom, I've got the mean rate of bubbles, or actually the rate of photosynthesis up the side, and I've plotted those, and you'll see as light intensity increases, so does the rate of photosynthesis, or the number of bubbles produced. But you get to a certain light intensity and actually the rate starts to level off.